As we age, our genome remains largely unchanged in its sequence, but its expression becomes increasingly dysregulated. This phenomenon, referred to as epigenetic drift, represents the gradual, stochastic alteration of epigenetic marks such as DNA methylation, histone modification, and chromatin remodeling. These changes influence which genes are expressed or silenced, regardless of DNA sequence, and over time, they can subtly or profoundly alter cellular behavior. One of the lesser-known but highly consequential outcomes of epigenetic drift is its impact on autophagy, the essential intracellular recycling process that maintains cellular integrity. Disruption of autophagy due to aging-related epigenetic changes has wide-ranging effects, not only on cellular health and longevity, but also on cancer risk. Autophagy is tightly regulated by a network of genes and signaling pathways, including ATG, autophagy-related genes, ULK1, BECLIN1, LC3, and regulatory systems like mTOR and AMPK. The expression of these genes is epigenetically controlled to respond to environmental cues such as nutrient availability, stress, and inflammation. With aging, this regulatory landscape becomes increasingly disordered. Aberrant methylation of promoter regions in autophagy genes, for instance, can suppress their transcription, reducing the cell's capacity to initiate autophagic flux. Similarly, alterations in histone acetylation patterns can lead to chromatin condensation around autophagy-related loci, silencing genes essential for cellular maintenance and stress resilience. This decline in autophagy has serious consequences. Damaged organelles, especially mitochondria, accumulate, generating excess reactive oxygen species, ROS, and triggering a cascade of further DNA damage, cellular senescence, and chronic inflammation. These conditions not only accelerate aging but create a microenvironment conducive to tumorigenesis. Cells that are unable to clear damaged components or respond to metabolic stress appropriately are more likely to enter a state of dysfunction, or worse, transform into cancer cells. The silencing of tumor suppressor genes via age-associated epigenetic drift compounds this risk, creating a perfect storm of genomic instability and impaired quality control. Moreover, epigenetic changes in aging cells can affect not just the basal function of autophagy, but also its inducibility. In youth, cells can upregulate autophagy in response to caloric restriction, fasting, or oxidative stress. But with age, this plasticity is diminished. Even if external stimuli promote autophagy, aged cells may fail to respond adequately due to epigenetic barriers that lock key regulatory genes into inactive states. This inability to mount a robust autophagic response under stress contributes to tissue degeneration and increases vulnerability to age-related diseases, including neurodegeneration, metabolic dysfunction, and notably, cancer. In cancer, the relationship becomes even more layered. Some tumors exploit autophagy for survival, particularly under the nutrient-poor, hypoxic conditions of the tumor microenvironment. But aging-related epigenetic suppression of autophagy may paradoxically promote the initial development of cancer by allowing damaged cells to persist and proliferate unchecked. This dual role, autophagy as both cancer suppressor and cancer enabler, means that aging-related epigenetic drift can shift the balance in harmful ways, first by impairing prevention, then by reducing control over tumor progression. Therapeutically, this opens up a critical avenue of intervention. Agents that reverse epigenetic silencing, such as histone deacetylase, HDAC, inhibitors, or DNA methyltransferase, DNMT, inhibitors, are already under investigation in cancer therapy and may have roles in rejuvenating autophagic function. Nutritional and lifestyle interventions like intermittent fasting or exercise, known to influence epigenetic patterns, could also help restore autophagic capacity in aging tissues. These strategies aim to reinstate the youthful responsiveness of autophagy, both to prevent cancer and to enhance treatment efficacy in older individuals. Still, manipulating epigenetic regulation is inherently risky. These marks are context-specific, and their systemic alteration may lead to unintended consequences, such as activating proto-oncogenes or impairing essential cellular functions. Precision is critical. The goal is not global epigenetic reprogramming, but selective rejuvenation of autophagy-supportive pathways in tissues most at risk for age-related decline and malignancy. In summary, epigenetic drift with aging gradually rewrites the regulatory instructions for how cells maintain themselves. When these changes interfere with autophagy, the effects ripple through every level of cellular health, increasing vulnerability to cancer and accelerating systemic aging. Understanding and intervening in this epigenetic autophagy axis may offer one of the most promising and potentially reversible targets in the fight against both degenerative diseases and age-related cancer.